Hello everyone and welcome to Samiki. My name is Giovanna Brado. I was advised by Professor Vanessa Santos Sotomayor and I study biotechnology at PUC-PR. My project was named as BCL2L1 in colorectal adenocarcinoma, where my job was to analyze and interpret data about the expression of BCL2L1 in colorectal adenocarcinoma, and then write a scientific article about it. This project was approved by the Ethics and Research Committee of Hospital de Clínicas da Universidade Federal do Paraná. So, colorectal cancer is a malignant neoplasm that affects two portions of the digestive tract, the colon and the rectum. And although the molecular bases of this cancer are actually well understood, there's a lot more we can do to better understand different molecular subtypes and help medical professionals to choose the right treatment for the right patient. BCL2L1 is actually an oncogene that has two isoforms, one of them being BCLXL, the large isoform. BCLXL has a anti-apoptotic ability as it binds to Bax and Bach protein and inhibits the activation of caspase cascade. So, therefore, no apoptosis and the cancer can survive. So, all the steps were done by my professor's study group before my project started. They gathered 73 cases of colorectal adenocarcinoma from Hospital de Clínicas da Universidade Federal do Paraná and used tissue microarray to choose different tumor areas such as superficial, intermediate, and profound invasive front areas. They use immunohistochemistry to detect BCLXL expression and use color morphometry to evaluate expression. Then they did a statistical analysis on the results. Here's an example of a photo of color morphometry, where red areas are immunopositive areas for BCLXL and yellow areas are immunonegative for BCLXL. So the results. We've seen that BCLXL expression was higher in control samples than neoplastic ones. It's important to point out here that most of our control samples were found tumor borders, so it's important to remember this when interpreting this data. BCLXL expression was also higher in patients with compromised lymph nodes. BCLXL expression was higher in patients that survived after surgery, and we've seen that there was a positive correlation between BCLXL and Parkin, which was unexpected because Parkin is a ubiquitin that sends BCLXL to proteasome degradation. So the discussion. BCLXL have an important role in cancer survival and proliferation. Higher expression on control samples showed us that the BCLXL promotes survival of newly mutated cells. And furthermore, BCLXL was identified mainly in patients with lymph node metastasis, so it's important for its proliferation as well. The mechanism by which BCLXL is associated with patient survival is still needed to be determined. However, similar results were seen with melanoma and renal cancer before. BCLXL expression with Parkin might be due to a PARK2 mutation, as PARK2 is the gene that codifies Parkin, and a mutation in the gene might affect the ability of the protein to bond properly to BCLXL. So my article was named as APEX1 and BCLXL expression in colorectal adenocarcinoma, and it's being revised and will be soon submitted to GCO Precision Oncology. I want to thank Professor Sotomayor and her study group for the guidance and support. Also, I want to thank Fundação Araucária for this opportunity, as well as for you for listening.